signaling. One is long distance, which uses hormones. In endocrine signaling, hormones travel through the circulatory system in animals. However, in plants, these hormones would travel through vessels. The other type of cell signaling is local. Paracrine signaling is local signaling in animals. This is important because local regulators stimulate target cells to grow and divide. Synaptic is a specialized type of local signaling in the nervous system, such as a neurotransmitter that stimulates target cells through electrical signals. Quorum sensing is when bacteria can sense local density of cells to coordinate their actions. Lastly, cell-to-cell is prevalent in embryonic development and immune response. Here, cells have direct contact.
Mating is an example of the signal transduction pathway. Cell receptors receive opposite cells' mating factors. This phase is reception. Next, transduction receives the signals and the mating factors fuse together. The final stage is cellular response, where the mating factors form a new cell.
This is a diagram of signal transduction pathway taking place in the cytoplasm of a cell.
During reception, signals sent out are heard only by its respective mates. A receptor protein in the target cell allows for the cell to hear the signal, in which the signaling molecule is specific in shape and site to attach to the receptor. The signaling molecule acts like a ligand, specifically binding to another receptor, causing the receptor protein to change shape. This shape change activates the receptor, allowing for interactions among other molecules.
After reception, the signal is changed into a different form, usually through a shape change of proteins in transduction. Calcium ions act as second messengers, which help to transport the signal quickly. Since cells can regulate its concentration, calcium ions can control the strength of the cellular response. Once the signal has been transduced, the cell can then regulate one or more cellular activities. The cellular response usually occurs in the cytoplasm or nucleus of the cell. The combination of proteins in the signal transduction pathway leads to a very specific cellular response.
Cells enter apoptosis when they are infected, damaged, or at the end of their lifespan. Apoptosis is when the dying cell is trapped and packaged into vesicles which are digested by scavenger cells. This protects neighboring cells from enzymes being leaked out of a dying cell. When the dust signal is not received, a protein inhibits apoptosis from occurring. When a dust signal is received, proteins are activated, which causes the cell to form blebs. This process is very important in shaping organs during embryonic development.